Thank you, Jeremy, for the kind introductory words, and thank you, Anna, for giving me this opportunity. So what you see on the screen is an image generated with DALL-E 2 from OpenAI. It's a pretty cool instrument, and this other picture is also from DALL-E 2. So trying to imagine how drugs could be discovered by a computer, and you put in the text, and if you don't like the image, you just change the text and you come up with a new image, or actually 4 images it suggests. So it's a pretty cool tool. So I think one of the answers, can humans learn from machine learning in drug discovery? One of them would be inspirational images, and I think we can learn from DALL-E 2 in this respect. First, I would like to focus on applied machine learning on target disease knowledge graphs, and this is work done at e-IDG. But before I do that, I want to put things in context. So there's been a revolution in text mining and the way we process large corpora of text. This comes from Tomasz Mikolov, who was at the time at Google. He came up with a way to vectorize word representation, and he famously solved this equation: king minus man plus woman equal queen. And this literally has improved our text-to-speech recognition technology dramatically, to the point that our phones can now transcribe text as we speak, or literally any text, and there's multiple language translators that work really well. You can buy them, you can use them. It's really amazing. For those of you who like Star Trek, there's even a Klingon translator online. The way this has been used, my colleague Lars Jensen in Denmark has been using BioBERT, which is one of the language transformers that's based on this technology from Tomasz Mikolov. It literally is a system that Lars has developed over the last decade or so, that every weekend goes through PubMed and uses named entity recognition and tags diseases and proteins, and it looks into full text, and it also looks in abstracts where full text is not available. And it allows us to come, for example, with this really cool application, which is this fractional publication count. So it looks at the co-occurrence between disease terms and protein names or gene names. And it has 3 channels: text mining, knowledge mining, and experimental channel. And on the experimental channel, in the diseases 2.0, Lars has decided to use an IDG tool called Taiga, which was developed by Jeremy Yang at the University of New Mexico. And you probably heard a talk from Taiga in an earlier version of the e-IDG Symposium. So just to give you a flavor where this goes in terms of machine learning, the possibilities are literally endless, but the trick is, of course, to try to map different bins and put like with like. So for example, expression data goes with in the bin of expression data. Pathway information goes into a separate bin, protein interactions separately, etc., etc. So it all goes into a database, which at IDG is called TCRD, and then that ends up into a knowledge embedded graph. And then based on a technique developed by Daniel Himmelstein while at UCSF, which is degrees weighted path connectivity, it looks at the way different nodes are related. So by nodes, we mean phenotypes, genes, diseases, and then the edges are the relationships between those nodes. And then we built a machine learning model where we fed the system a number of genes associated with Alzheimer's disease. And we predicted about 16,000 diseases and 16,000 proteins. And of course, just to put things in context, despite the fact that we're experiencing COVID, we all suffer from the possibility of Alzheimer's later in life. It's quite a serious disease. It's still one of the primary causes of death. This is despite the fact that just about 120 years ago, it was not a recognized disease entity. So it's a fairly new addition to the medical dictionary. So going back to the machine learning, when we fed the model and we predicted the 16,000 genes, we got a list of genes that were ranked. So I went to the group of Kiran Bhaskar at UNM and said, here are the top 20 genes predicted, which are on the left side. By the time we submitted the paper to its third revision cycle, one of the reviewers said, you actually should look at the negative ones too. So we also looked at the bottom genes as well. But just the image here suggests that inflammation is one of the critical pathways associated with the top 20 genes. There's ER stress, some metabolism, signal transduction, etc. So once the experiments were conducted by Jessica Binder in the Bhaskar lab, what we found was that among the different types of experiments, of course, not all genes were ranked equally, and not all genes were confirmed in each of the experiments. And this is somewhat consistent with what we found in the paper. So when we validated the machine learning model with an external set from Lars Jensen's diseases with 200 genes that we selected as a prediction set, we found that it correctly predicted the 20 genes that we knew were associated, and also correctly predicted some of the negatives. But in predicting the positive genes correctly, we also found that only 1 in 3 of the predictions was correct. So the chances of having a good prediction was 1 in 3. Hence, we looked at 20 genes on the top list. So it's not surprising that only 5 were validated. And what's described also in the paper, so one of the genes is a dark gene, so genuinely a novel association, but what we note is that there is a strong relationship with innate immunity pathways, and that's probably a critical component of Alzheimer's disease. It's been studied, even the group of Joel Dudley at Mount Sinai published a paper where they found herpes virus infections in the post-mortem analysis of Alzheimer's patients. We also found in the bottom genes some that suggest a relationship with Alzheimer's disease. This is partly our learning of the way machine learning works. So to address the question, can we learn from machine learning? The answer is yes. We looked at the top 20 genes, but what we didn't realize in considering is that when you feed the model that just has a 0 and 1, then just because the model ranks the genes by the probability, any probability that's above 0.5 should be considered as equal. So in reality, when we wanted to look at negative genes or bottom genes, we shouldn't have looked at the lowest ranked, which are potentially still associated with Alzheimer's. We should look at those in the middle that have 0 association. So that was one of the learnings we took from this. The other learning that we took is that initially the paper was rejected in Nature Communications and other top-tier journals because we didn't find anything related to the amyloid precursor protein or APP pathway. And as you probably have seen by watching the news, a lot of phase 3 clinical trials targeting APP with monoclonal antibodies have failed. It turns out that a lot of those antibodies were predicated on the promise of APP based on a paper by Lenné et al. In Nature 2006. And an interesting investigation published in Science very recently suggests that some of the data that was associated with those papers was essentially doctored. And had we known about this, we probably could have used it as an argument, because we literally state that our model suggests that infection might be the cause of Alzheimer's. And so this is where machine learning doesn't bring a bias. The only bias that it brings, of course, is the initial set of genes that you select or the initial training set. Of course, there is bias in that. And then, of course, the other bias would be how do you interpret the model? But if you really look at the model and try to understand what it tells you, in this case, it was telling us that infection might be the cause of Alzheimer's. So at least I put that claim out there that machine learning suggests Alzheimer's is caused by infection. So I'm going to switch gear and tell you something from our work at Roivant, which is primarily focused on small molecules and the application for machine learning. The major effort that was requested from us was to develop tools that would rapidly allow using MLflow and so production grade machine learning to analyze billions of molecules in real time and predict them across hundreds of models. And this is just a sample of the models that we built. So for example, kinetic water solubility, membrane permeability in a cell line called MDCK, where you look at apical to basal versus basal to apical. So these are, of course, different models. This is measured separately. There's HERG, which is a cardiac toxicity related ion channel. And the idea is, of course, to develop tools that build upon that. And what's critical for this type of analysis is to build models that are predictive. So we put a lot of effort in making sure that on molecules that had not been seen by the model, we are within 1 log unit, which is consistent with the experimental error for those assays. So whether you look at MDCK permeability or KCO2 permeability, human hepatocytes, the only one that we're not doing really well is human liver microsomal stability, where a lot of the molecules that are predicted stable were in fact measured unstable. We are, of course, applying course correction to try to understand what goes on with that model and try to figure it out. I think we begin to understand that part of the problem is assay compatibility and trying to figure out stuff that works. So what I learned from machine learning is that sometimes you look at large data sets and they're not very useful. So this is a water solubility data set published by the Burnham Institute back in the molecular library days, almost 60,000 compounds. Using a chemiluminescence assay, we try to make heads and tails of this. It's not really useful. So the data set doesn't work. You can't use machine learning. You can't even confirm known drugs that are in the data set. If you look at their measured solubility in this assay versus other measured solubilities from different sources, they're simply not compatible. So this data set is useless. The other thing that we had to learn in the process is to listen to what the chemists were telling us and understand what they actually needed. So initially they said, we want a model, a machine learning model for water solubility. It took us about 2 months to figure out that what they really wanted was a model for kinetic water solubility as opposed to thermodynamic water solubility, which is what most people would assume when you look into the literature. So we spent significant amount of time chasing the wrong metric. Luckily, we found in the NCATS ADME predictor dataset a set of compounds that is compatible. So the assay is compatible with the assay that was used by Roivant. So we were able to build a model that is reasonably predictive. So the really cool feature of this was that just using chemicals from the NCATS dataset plus the 2021 chemicals from Roivant, we were able to correctly predict soluble, not soluble for 84 out of 87 of the newer chemicals. And the other model that I was going to spend a bit of time. So I told you about MDCK. This is a dog colon cell line. Where they look at permeability. On the surface of it, if you look at literature, it suggests 24,000 data points. After you massage them and clean them up, you are left with far fewer, so like at least an order of magnitude less compounds. And of course, you have to exercise care and build models separately. So in the literature, they use MDCK, which is the native cell line, but they also inject a human gene for the ABCB1 transporter. So those are separate. It wouldn't be the same assay, and the data aren't exactly compatible. So this literally happened to us a few months ago. We had built a model, and then external prediction on compounds published after 2019. So the data set was trained on 2019 and prior, and then 2020 and 2021 were used as prediction. It looked really solid. We replaced the data with internal data from Roivant, and it still looked solid. So we were really happy. And then what we really found is that we were using the Y variable as one of the descriptors. So of course the model would work all the time. Once you correct the model, of course, it doesn't look that good. The explanation for that has to do with the fact that the chemical space of the molecules from Campbell is very different from the chemical space from Roivant. So of course, the solution for that is to inject some of the molecules from Roivant, so for instance, the 2021 into the training, and then you predict the 2022. And then once you do that, the prediction is, of course, within 1 log unit, which is pretty good when it comes to qualitative evaluation. So the lessons that we take from doing production grade machine learning is that you really need data that is compatible with internal company data, and you can't just keep using literature data to build models, because the problem is not the algorithm. The problem is having good quality data that you feed into the model. Otherwise, you end up into the garbage in, garbage out scenario. So the best path forward is, of course, to find that subset of the literature which is complementary with your data set and ensure that you have both assay and chemical space complementarity. So the other learning, if data looks too good to be true, it is too good to be true. So you have to at least hunt the error, see if you find it. So in our case, so Christian and I had been doing machine learning for 30 years each. So we still took a day to figure out what the problem was, and Christian, of course, figured it out soon enough. So trust but verify should be a motto for anything related to machine learning. The other question, of course, is when you look at production grade, if you build the models on a platform, but then deploy them on a different platform, which is our case, the models were slightly different, and you have to be aware of platform dependence and other things like that. So I'm going to spend the last part of my talk discussing the other side of computational chemistry. Most of you associate computational chemistry with quantum mechanics or molecular mechanics. I'm just going to mention a retrosynthetic analysis based on Drug Central. The paper has just been approved. It should be out online in a few days. But we started with technology that was built by Bartosz Zubowski and his team at Alchemy. And they've made the rounds with high impact publications with asking really relevant questions about the chemistry of life or sustainability in the current environment. So what we did, and this is really all of the work, the rest is just the visualization. We started with Drug Central. We focused on small molecules, which is about 4,200 drugs, a bit less than that. The majority of them are approved in humans. So the newest version of Drug Central has veterinary drugs as well. And then it turns out that about 2,000 of these drugs you could make in up to 7 steps, starting from 100 of the most popular reactions. Another 2,000 are doable, but you have to expand that to about almost 2,000 reactions. So that, of course, we're in the process of reorganizing and reevaluating to minimize that, because the goal is to build generative chemistry tools. And then about 100 are so-called rare scaffolds. So they're not present in the Zinc database or some other situation. And one in particular was declared as unable to synthesize. It has to do with this hydrated keto group, or a ketal. This is entirely unstable, so this would basically leave a water molecule and would turn into a keto group. However, the FDA approval leaflet for this drug approved last year shows exactly this chemical structure. So we think they found a way to stabilize it, or it forms the hydrated form under certain conditions in the human body. So that might be the active metabolite. So some of the questions we asked to try to understand this, if you look at the approval of drugs, the obvious question was, have this synthetic accessibility changed over time? The short answer is not much. And it turns out that there was no dramatic change over time when it comes to that. Yet at the same time, we were aware that a simple descriptor like for size, which is molecular weight, if you simply look at molecular weight, you see very clearly that drugs do get heavier over time. So if you just follow the really sky blue part of the slice gets smaller and smaller, and then the darker ones get bigger, which means drugs get heavier over time. So then the question was, how else can we investigate this? The answer is you can simply look at the number of starting materials that are used to build the drugs. So if you put them in as a string of ingredients for the chemical reactions, the answer is very clearly that initially most of the drugs were made with 2 ingredients or 3 ingredients, but then over time the number of ingredients increases all the way up to 15. So there is a shade that goes from yellow all the way to total black, and you can see that trend quite nicely. So the other question was, can we actually see how this evolved over time when it comes to chemical process manufacturing? So 10 pharmaceutical companies have put together what's called a PMI score, which is process manufacturing intensity. It looks at the mass of the product versus the mass of all the reactants that go into the reaction. And then you have to normalize that by the number of synthetic steps taken for that. So that gives you a measure of synthetic complexity that is not calculated, but rather based on actual experiments. So the way that indicates is that over time, synthetic complexity has increased quite dramatically, and that's what's shown here. I wouldn't go through the numbers because they're arbitrary, but they show a clear trend that PMI divided by path length has increased over time. One really cool thing about this particular slide, I was attending EuroQSAR in Heidelberg, and I talked to Lutz Weber, who works at Ontochem, and he literally generated the first time to patent for all drugs in the space of less than 48 hours. So I was able to add this slide for that talk. So just the conclusions based on this retrosynthetic evaluation. It looks, if you look at synthesizability, molecular weight definitely increased over time, as did the number of starting materials and synthetic complexity. It sort of stands to reason to imagine that the complexity of reagents that have been available over the last 8 decades has also increased over time. But I think the general conclusion that drug synthetic complexity has increased over time stands regardless of that. And that probably is an evolutionary demand due to the growing intellectual property landscape. So people need to patent. And of course, the only way to patent is to come up with something novel. So we're approaching the end of my talk. So instead of conclusions, I think the future of artificial medical intelligence is upon us. Hopefully, we will soon be able to solve the equation human minus health plus disease equal patient. And then, of course, to reverse that. So we would go back where we would do patient minus disease plus health. So we should be able to figure that out using combined data set with omics, biomarkers, electronic medical records, etc., in real time. And one easy application that I can think of is if you have a smartwatch and it tracks your health status, it can inform Alexa what you should be at least encouraged to eat on that particular day. to keep your health status. So looking forward, the way it looks in 2022, I resonate with this paper that was published, or an article that was published by Iulia Georgescu in Wired magazine that argues that humans shouldn't be the sole keepers of scientific knowledge. So we could imagine an AI that stores all the information. Of course, the trick here is to have an improved domain expertise, which I think is really the future. It's how we would visualize the future. So having academia and pharma partnerships working together to validate computational models, and then trying to rethink computational medicine on that basis. So in my opinion, the golden era of machine learning is upon us. And then last but not least, I think it takes a team to discover drugs. The eureka moment will no longer happen to a single person. I think at least with drug discovery, it will really take a team. And the trick is, of course, to apply translation informatics to articulate that information, to generate domain-specific scientific knowledge and integrate with real-world evidence towards what's called a hybrid human AI model. Also, to let everyone know, this is my last public talk as part of Roivant. So as of November 30, I will no longer work at Roivant. They laid off 100 people, and our entire team is part of that. So we're exploring new opportunities. So with that, hopefully I'm on time, and I'll stop sharing.